My name is Martín Rottemberg. I will present you a collaborative project that deals on how immune responses control intracellular parasites and bacteria. I am professor in infection and immunity at the Karolinska Institute in Stockholm, Sweden. I come originally from Argentina. So the project is more specifically deals with how T cells, one of our main immune response components, control infections with human intracellular parasites. The project is coordinated by Professor Pilar Aoki. She's at the Department of Chemistry at Córdoba National University. This is Pilar, and it's also coordinated by Miriam Postan, which I have known for many, many years. She's my friend, and she's at the Instituto Nacional de Parasitología in Buenos Aires, so National Institute of Parasitology. And this is Miriam, and by me. So it is with two different infections that share common biological features. One is Chagas disease, and Chagas affects around 6 million people in Latin America, and this is a chronic disease, so it's lifelong and can span from symptom-free to life-threatening. Chagas is caused by the infection of a parasite, of a protozoan parasite. This is this one. And it's called Trypanosoma cruzi. And this parasite is transmitted to human beings by a fly called vinchuca. And once in our bodies, the parasites will infect different types of cells, and within these cells, they will multiply. So here you see this is one cell, and here this is a nucleus of a cell, and here all these dots here are parasites that have actually multiplied within the cell. So we are working with another infectious disease that is tuberculosis. And tuberculosis caused by infection with Mycobacterium tuberculosis affects around 25% of people worldwide, and more than 10 million people develop tuberculosis yearly, and more than 1.5 million will die every year. You can compare this number with those that have died with coronavirus, and the tuberculosis doesn't get the same press, probably because it infects people, the incorrect people from poor countries. So it's poverty-related disease. Again, this tuberculosis is caused by infection by a bacteria, it's called Mycobacteria, and is transmitted via aerosols. So a person that has tuberculosis, individual will cough and will generate like aerosols, and these will be inhaled by a healthy individual that will become infected. Of course, the tuberculosis infection can be is usually located in the lung, but can also be located in other sites of the body. And this is how the lung will look like. So this is a typical lesion produced by tuberculosis. So again, tuberculosis as Trypanosoma cruzi is intracellular, so it will infect different types of cells, mainly phagocyte, and then it will multiply within this phagocyte. So So again, this is a phagocyte, or in case of Trypanosoma cruzi, it can be or whatever cell. And here you have the pathogen. In this case, Mycobacteria. The same is true for Trypanosoma. And these parasites, these pathogens, will multiply happily in the infected cells. Here you see how this is how it looks in the microscope. I don't know if you can see. We have some dots in purple here, and those, in this case, are bacteria. Now, when you have an infection, you have an immune response, and the main component that protects and reacts against this infection are T cells. And T cells will recognize specifically these infected cells and see that that infection within these cells will be controlled. So what we are studying is how these T cells are regulated, and so we even working with different infections, which we have shared interest in features of this T cell mediated control. So we have a program. We have a small collaboration that is sponsored by a Swedish funder. It's called Stint. And we have both research and education programs within our collaboration. So we plan to exchange materials and personnel. We plan to visit each other, perform seminars, workshops, teach in each other's sites, and so forth. So thank you very much.